All right, back again. Doop-a-doop-a-doo. Okay, so kind of getting back to that point that I was making just before we left, it's all about how many alleles you have for a given gene, right? And because we're diploid, right, for all of the autosomes, we should, pending any issues with meiosis, have two potential copies. Well, yeah, we have two copies of each allele and potentially two different alleles, right? And so that means that it's somewhat rare to have two recessive alleles for any given gene, right? It's obviously not completely rare, but it's somewhat rare. Now, in genes that are on sex chromosomes, now you can have the situation where you only have one set of alleles for all of your genes on that chromosome, right? And that causes a couple of things to happen. One, and I'm gonna blow this up a bit because this is teeny tiny. It causes what are called non-reciprocal crosses, right? And so really what I want you to get out of this is that normally, and this is kind of one of the Mendelian principles, right? It doesn't matter which way round you do the cross, whether the female is homozygous dominant and male is homozygous recessive, or vice versa. It shouldn't make any difference. But with sex-linked traits, it does. And the reason for that is how these alleles kind of pass through those generations, right? So if we were to do a monohybrid cross, but now we have genes on the X chromosome, like the white gene, which results in white-eyed males, it matters a great deal which way round you do that cross, right? So essentially a white-eyed male, right? And it's white-eyed because it has one recessive allele, right? That only contributes one kind of mutant chromosome to the next generation, right? And you'll see that here in this heterozygous female, right? And so because from the female side, you'll have a normal X chromosome and you'll get the Y, so you'll have a red-eyed male. This female is now called a carrier, right? And carriers are, and this is kind of a little bit of a sex linkage term. They're heterozygous females. So they carry the trait, but don't express it. Right, because they're heterozygous. It's a recessive trait. Now you'll see that recessive allele pop back out and you'll get a white-eyed male, or 1 1/4 of your offspring will be white-eyed males. Now, if you look at it the other way around, now you have two white alleles, right? Because you have a white-eyed female. Oh, she liked her bone. And now each of the F1 generation will carry a copy of that allele. Again, you'll have a heterozygous female carrier. So she will be red-eyed because she has a dominant allele, but she'll carry the white allele. And now you have a white-eyed male because that individual inherited one of the two mutant X chromosomes from the white-eyed female. And so when you cross those together, you will get, instead of a 3-to-1 ratio of normal to recessive, now you get a 1-to-1 ratio. So that's one consequence. There's my... Oh, she's giving her a bone. Or she's getting sausage as well. Oh, it's trick the puppy time. The other consequence, good job, is that you will have traits which are expressed much more commonly in males than in females. Can anybody think of, in humans, some of those traits? There's some real well-known ones. Color blindness, cool, that's good. So you can't see, I think, red and green. I think that's somewhat common in males, very, very rare in females. What else? Hemophilia, cool. That's a good one too, Maria. There's another one too, which I can think of. There are probably more than this, but this is these are the three that I can always remember. Boldness, right? Or male, specifically male pattern baldness. That's why you see many, many, many more men go bald than women, which is kind of neat. So this is like a clear characteristics of a sex-linked trait. Obviously, if you're not able to do these reciprocal crosses, now instead what you do is you look at a lineage instead. And this is, I'm going to again blow this up a smidge so it's easier to see. I just had to fit it onto the actual page. Right, so this is back to old Queen Victoria and Albert. There's actually a museum in London called Victoria and Albert Museum, which is a pretty cool one. Anyway, this is the Padovan inheritance of hemophilia in the European royal family. Here we've got our Tsar Nicholas II of Russia and his wife Alex. Right. Alexis, that's it. That was his name, Alexei. Right. So here, this is like a standard nomenclature, right? This is a standard way of writing these family trees where females are round circles, males are squares, and then a full colored circle or square indicates the expression of a disease, right? So typically a person having the disease phenotype, and the half-colored well, they're going to be circles if it's sex-linked, are carriers, and so these will always be female. And so if you look at the European royal family, all the way from Queen Victoria, who was a carrier of hemophilia, and she must have, actually, how did she get it from? Maybe it was a spontaneous mutation. But either way, Jordan, question.
Sorry, is this a guess, or is this just like people have approximated just from knowing that these children were
this is known.
Positive for this? I was like, I didn't know. Did they do DNA on corpses or something? I just my curiosity.
No, hemophilia, at least before the 20th century, was pretty much a death sentence for individuals, because you would basically bleed out.
Yeah.
I think I've actually gone through and I can't remember all their dates, but I think the oldest that any of these have survived is, I think, like 30, give or take, 35, something like that. So yeah, so this is the fun thing about these family trees, is that you can actually figure out the genetics from the individuals that have the disease, right? So the individuals that have the disease are either homozygous if it's autosomal or hemizygous recessive, right? Because they're expressing the disease. And so from Alexei, which was the doomed child of Tsar Nicholas II and Alex, he was a hemophiliac. So you know that his mother must have been a carrier, because that's the only way that he would have gotten that allele. He wouldn't have got it from his father, because his father, if he'd had it, would have been a hemophiliac too. Most likely would have died before reproducing. So you can actually work backwards. It's kind of fun. It's like a logic puzzle, and figure out who has what. Sometimes you can't be 100% sure because it relies on those individuals having offspring. So we'll never know whether or not Marie Anastasia or whoever, these 4 children of Tsar Nicholas, 4 female children, sorry, whether or not they were carriers or not, because they never lived long enough to reproduce, and we can't figure it out from their offspring. But for all of the individuals that did have offspring, we can figure out whether or not they were carriers or where that allele came from. Because you just work through it. It's just a puzzle, basically. And so you can see here, obviously, who is that? Edward VII didn't get the recessive allele. It's actually it's a blood clotting factor that activates thrombin in your blood, which results in forming a mesh over wounds or whatever, which stops blood leaking out. We know that Edward VII didn't have that allele because he was not hemophiliac. So all of his offspring, even though they kind of joined together again down here, which is super weird, all of his offspring and descendants don't have hemophilia. The same with Victoria II, I'm guessing, or she would have been if she'd been queen. We know that she wasn't a carrier because her son Wilhelm II did not have hemophilia. So it's basically you go through and you work through it like a logic puzzle. It's kind of neat. And so the key thing that stands out here is that only males have the disease. There are no females with a full red circle here, because the only way a female could get hemophilia is if she was the result of a hemophiliac male siring child with at least a heterozygous carrier female, which is very, very rare. And so this is a classic trait, and essentially, you know that it's recessive because it disappears as well, right? So no one in this generation that we know of had hemophilia, but now we have Leopold having hemophilia in her offspring. She had loads of kids. What's that? 1, 2, 3, 4, 5, 6, 7, 8, 9. Wow, that's a lot of children. But you can see that trait kind of continuing on, right, down through generations, and it has to go through a non-carrier for it to disappear. So this is a classic sign of something being sex-linked, right? If you only, hey, get out of it. Don't need to see that. If you only see it or predominantly see it in males. Right, and so you just go around, go to Walmart or, a football game or whatever, and just kind of look around. You'll not see almost certainly any bold women, but you'll see plenty of bold men. And you can follow that boldness back through generations and essentially see where it came from. So I know that my uncle on my mother's side, he was bold, really bold. And he must have gotten that allele from my grandmother, yeah, who must have been a carrier, because my mother didn't get that allele and didn't pass it on to either of my brother or I, because we probably would be expressing it by now if we did. So it's kind of fun, right? You can just kind of work this stuff out. And so also that means for sure that yeah, that means for sure that my grandfather did not have male pattern baldness, even though I know he didn't, because he had like loads of hair. But you can work it out through, hey, quit it. First principles, just by working your way back up through the generations. It's pretty neat. I like doing this. Anyway, any other questions on sex linkage? Okay, so just to kind of wrap up this little branch of oddities, just a little bit about the Y chromosome. And the Y chromosome is super odd. There are very few genes on it. It's very small, like really small compared to the X chromosome. And it doesn't undergo recombination, because what would you recombine with, right? You cannot undergo recombination between the X and the Y because they're vastly different in size. But they need to stick together during mitosis and meiosis, so you get normal segregation. So they do have these kind of like weird regions at the ends near the telomeres, which quit scratching, which are there to allow those chromosomes to line up during cell division. You want to get down? Yeah. Sorry, a little bit of puppy time. It's very cute. Now, because they don't undergo much recombination, you don't get very much shuffling, right? So all of our autosomes and even the X chromosomes in females, hang on. There you go, batty dog. Off she goes. Will undergo recombination. So whatever combination of alleles you got from your parents, those will get shuffled up a little bit. And so the ones that you give your offspring most likely will look a bit different. Not for all of your chromosomes, but for a bunch of them. And over time, those genes will, those alleles will kind of disperse and get all mixed up. That doesn't happen for the Y chromosome. And so what you get is a very distinct kind of lineage, right? And so you can get a very good male lineage from those, because really the only change that happens in the Y chromosome is via mutation, right? That's the only way to get new combinations of alleles, is to actually have mutation occur, which happens much more slowly than recombination during meiosis. And so you can actually use this to look way back. These are called haplotypes, right? Which is basically the collection of alleles on kind of one of your chromosomes or one of your sets of chromosomes. In this case, your Y chromosome. And so you can actually use that to go back to, shoot, when did Genghis Khan wander around? I think that was around 1100 AD, 1200 AD, I think, from memory. And so you can actually trace back, if you're living in that region, whether or not you're descended from, or presumably descended from, Genghis Khan. And you can actually see that that matches really well with the extent of the Genghis Empire, which stretched all the way over to the stands over here, Turkmenistan and Afghanistan and Uzbekistan.
Hey, Dr. Crook.
Hello.
Marcus here. Yeah, I was doing a report for evolution a few semesters ago, and it turns out that the haplotype that belongs between right next to Mongolia is
Huh?
from Neanderthal DNA, and people in Europe had a tough time with COVID because it made them more susceptible in some way.
Oh, that's cool. Yeah, I mean it's really weird because there's not an awful lot of genes on the Y chromosome, but that's not the same as there being no genes. And
Yeah.
so, um, yeah, this actually, when you start looking at kind of population level genetics, it's really, really interesting the kind of patterns that pop out. What's another one? Yeah, one of my favorite ones to talk about, I won't go into in great detail, is the CCR5 deletion in a T cell receptor, which confers resistance to HIV. And so that maps fairly closely with the frequency and severity of smallpox outbreaks in the Middle Ages.
Yeah, it was supposed to be with the severity, like for COVID, if you were likely to be hospitalized,
Hmm.
you were more likely to be hospitalized with that haplotype.
Yeah, it's really weird to think of our like who we are as not just the result of our parents, but also the result of, you know, potentially migration of people thousands and thousands of years ago. You know, it's also why Asians are quite frequently lactose intolerant, um, because the ability to tolerate lactose arose in the Eastern Mediterranean, stroke Middle East, around 6,000 years ago, give or take. And that spread mostly west. It didn't spread east, as those peoples kind of spread out through Europe and North Africa. So it's really cool to think about, you know, kind of like where we come from, essentially. And um, it's not just our immediate lineage, right? These are things that go back thousands and thousands of years. Yeah, that's actually an interesting point, Dipper, about lactose intolerant. It's actually one of the, you know, there's all these different theories about why um, certain countries or regions of the world rose to dominance. Um, yeah, Galen, and also Dipper. That's kind of what it. Basically, the chance mutation which resulted in lactose tolerance, which arose in kind of Eastern Mediterranean about 6,000 years or so ago. What it meant, so there was domestication of animals, right? So kind of proto cows and stuff. At that already by that point, my daughter's lactose intolerant as well. I don't know where she gets that from. Um, but basically, what it meant is that you could get nutrients and resources from cows without killing them. And so not only would you raise livestock to like kill it and eat it, you also get nutrients from that uh, animal before that happened, right? Because you can't just cut off half a leg of a cow, right? You got it's all or nothing. And so that's one kind of explanation as to why kind of European cultures became more dominant. It's obviously not the only one, but it's an interesting one to think about, though. Yeah, all kinds of weird stuff. Like earwax is totally different in Asia versus Europe. Totally different. Don't know why. Yeah, totally. Why is it in? Oh, I can never remember which way around. I have to think of my own ears here. Um, yeah, I can't. Oh, which way around does it go? I don't have enough of an earwax problem, probably too much information, to remember which way around. But Asian earwax is very, very different from European earwax. Why? Huh? Who knows? It's a mystery. Anyway, moving swiftly on. So this is really about talking about ways in which Mendelian inheritance, right? So transmission genetics kind of doesn't fully capture all of the complexity of what's going on, right? So these are really kind of extensions of transmission genetics. And so obviously the first one was, let me just go back, where'd that fly go? It's in it somewhere. Here you go. So sex linkage, right, was one of the very, very first examples of inheritance which did not follow Mendelian principles at the time, because he found a gene, the white gene, which was sex linked, right? Only or predominantly got expressed in male flies. And there are a bunch more, right? So really is probably a good an easy way to say that most inheritance doesn't follow simple Mendelian patterns, right? Mostly it's a mix of a whole bunch of other stuff, right? And so obviously the simple monohybrid and dihybrid cross ratios, the 3:1 and 9:3 ratios, can still be obtained, particularly for certain traits. But for many traits, or really most of them, you get other stuff going on. And there's a lot of reasons for that, a lot. So there are genetic reasons, right? So we've got something where we don't have a simple dominant versus recessive relationship. We have multiple genes determine the trait that you see, the phenotype, right? So this is called a polygenic trait, and most traits which you can see, like physically, in humans are polygenic. So eye color is a classic example. It's a result of at least 4 different genes, right? The sum, what eye color you have is the sum of all of those different alleles. Also with polygenic traits, now you can have funky stuff go on, which is like gene by gene interactions, right? Where those genes don't act independently. They influence each other. What do you want? I think she got scared by the rubbish truck outside. And obviously, sex linkage is a classic one too. Now, without a doubt, you are who you are, not just because of what genes you inherited from your parents, right? Also because of the environment in which you grew up in too, right? There's a lot of stuff which is very environment influenced, right? So we have this, what's called a genotype by environment interaction. Classic one is height, right? So, you know, I always use my own example. I'm quite tall. I'm 6'2". My brother's 6 foot. Both my parents were, I don't know, 5'8", if that, 5'6", something like that. And so it's hard to know whether they were just shorter, right, because of their genetics, or if it was because, I mean, they grew up in post-World War II rationing in the UK, right? You know, my dad didn't see an orange until he was like 10 or something like that, you know, in kind of the, what would that be? That would be in 1960. You know, so it's hard to know which, right? Because how tall you are depends not just on what genes you inherit or what alleles you inherit from your parents, but also how much nutrition you have available, the quality of that nutrition, and a whole bunch of other things as well. Jordan.
Hi, sir. I watched a video several years ago about epigenetics. How, I don't know, say how much evidence, but how is there anything you can trying to figure out how to word this? Is it real, or what is your opinion on it, or what have you
Oh, it's very,
heard about
very,
it?
very real.
Yeah, 'cause I'm like, I've only seen one video on it, so I don't know much about it except what I saw
Yeah, we'll be
6 years
talking
ago, I
about
guess.
epigenetics. Ooh, not today, I don't think, from memory. But we'll most definitely spend some time talking about epigenetics. Without a doubt, it is a fascinating topic, and it is very, very real, without shadow of a doubt. But yeah, we'll just have to sit on that one until we get to that point in the lecture series.
Gotcha. Okay, cool.
But yeah, we will get back to that without. That's also another fun thing, fun favorite of mine to talk about. Okay, so kind of now we get into talk about dominance, right? And so we've got a good, well, probably one of the few examples of dominant, incomplete dominance, which is in snapdragons. Pigmentation actually is the common one. And so really dominance is about interaction between different alleles at the same locus. And really it's making a statement about function as well, right? And so the shorthand is really that a dominant allele is a functional copy of that gene, and a recessive one is less so. But there's also more to that, right? So down here, loss of function mutation is almost always recessive, right? Because it just doesn't work. So if you have a functioning copy of that gene on the other chromosome, then you'll never see that recessive phenotype. That's kind of really what dominance is all about. But you can have other stuff going on. You can have a gain of function mutation, right? So that's where a gene or a protein may be functioning all the time, even when it shouldn't. Is that going to be dominant or recessive? Kind of depends, right? You can also have things called dominant negative mutations, right? So these are ones where that protein may stop functioning, but it has a dominant effect on whatever process it's involved in, right? So it's really kind of a lot more complex than a straight up dominant versus recessive deal. It really depends on context too. And also dominance isn't always an all or nothing deal, right? So sometimes we can get intermediates, right? Where, the heterozygotes are kind of in between the homozygous dominant and homozygous recessive, right? And really what this is talking about is that we have some degree of addition, right? So if you have no function at all, you have a certain phenotype. If you have a little bit of function, you have, kind of a partial phenotype. And then if you have more function, then you have the full phenotype, right? And this is what's called incomplete dominance. And it's very common in pigmentation, based traits. And so the reason for that is pigmentation isn't necessarily an all or nothing trait. And so you can have, and this is snapdragons is one example where you have homozygous dominant is red, homozygous recessive is white or ivory, right? And so essentially what you have to do is you have to mask that white color. And I always talk about if you're painting a wall, right, and you want to paint over a particular color, sometimes you have to use a lot of paint, right, to cover that color up. And that's really what we're talking about here in terms of incomplete dominance, right? So you get a certain level of function from each functioning allele. So if you have two functioning alleles, you have a lot of pigment, you have a deep color. If you have one functional allele, like in a heterozygote, you have some color, but you don't have enough to completely mask the background color of the recessive phenotype. Oh, hey, Maria.
Sorry.
That's all right. So what you see with incomplete dominance is the heterozygotes do not match the parents, right? Which is something that we weren't learned with mono and dihybrid crosses. That's one big deal. And the second is when you self-fertilize these heterozygotes, you don't get a 3-to-1 ratio. You get a 1-to-2-to-1 ratio because the phenotype reflects the genotype now. Right, so first thing is the hets or the heterozygotes, the F1s don't match the parents. And in the F2s, the genotype equals the phenotype. So we never do this with a dihybrid cross because that would be horrendous. But here you can see normally you have a 1-to-2-to-1 genotype ratio of homozygous dominant, heterozygous, homozygous recessive. But in a normal dominant recessive trait, you'll have a 3-to-1 ratio of the dominant to the recessive. In incomplete dominant, you'll have a 1-to-2-to-1 ratio because now you have a third phenotype, which is an intermediate between the two parental phenotypes. And there's also another example, again, pigmentation with aubergines, eggplants, called aubergines in the UK because it's French. And again, you have the same deal, right? You have an intermediate F1 and you have a 1-to-2-to-1 ratio in the F2s because you now have that intermediate phenotype. So I'm going to use a different example to the one in the lecture, just because in the PowerPoint rather, just because I like talking about blood type more. I think it's fun. But this is also another interesting way of looking at it too. So the level at which you assess that trait also matters, right? Generally speaking, it's easier to see simple traits at the kind of the gross phenotype level, like color, for example, or simple kind of obvious traits. If you start looking at the molecular or cellular level, you're more likely to see much more complex interactions between alleles, right? And so it very much matters what level that you're looking at this trait at. So the next few slides are good example. I'm just going to give you a slightly different one as well. Also, because this is fun. And this is something that, you know, you can relate to an individual level rather than still talking about bloody peas. You would have thought in like the last 150 years, 170 years, we'd get kind of like a more fun organism to talk about than peas. All right. What I'm going to talk about is codominance and blood type. And this is also cool because it's another example of you have more than two alleles in controlling a particular trait, right? And so in terms of blood type, you can be A, B, AB, or O. Right, those are the 4 different blood types. So I'm just watching my puppy go to sleep. It's very cute. Now your alleles, you can have A, B, or O. So how do we go from this, which is the alleles, to our blood type? Actually, let me just, you don't need to scrub this out. I'm just doing it because it's going to be a little bit easier to talk about this way around. And I call myself a geneticist, right? Should be thinking about the order of things. Right, so these are our alleles. And we can have a whole bunch of different genotypes. From those 3 different alleles. Right, so those are the genotypes. But now we can have essentially the blood types are phenotypes. Now, most of this works in the classic dominant recessive fashion, right? So if you have a dominant A allele, you're either homozygous dominant for it or you're heterozygous with the O allele, you'll have blood type A. And I'll get into how this actually works in a little bit. Same goes for homozygous dominant B or BO heterozygote, you'll be blood type B. And obviously if you're homozygous recessive O, then you'll be O. Simple as that. The weird and funky bit is if you are heterozygous, but you're heterozygous for the 2 dominant alleles, you will express both of those alleles in your phenotype. A is not dominant to B and B is not dominant to A. Jordan.
Hi, sir. I thought O was like the most common blood type. Is it just that there's a lot of people who?
I don't know. I don't think O is the most common. O blood types, they're in huge demand because they're the universal donor.
I think it's for O neg, but I guess O pos works for like men. It's just that, you know, pregnant women or women who have children becomes an issue, I guess.
Yeah, the rhesus factor is a big deal for pregnant women, but for everybody else, just O is the universal donor. I'll get into why in a second, which is kind of neat.
Gotcha.
So for AB, you express both of those alleles, right? And so kind of the question is how, right? How do you have that? How do you have 2 different alleles be dominant at the same time, right? Because that doesn't really jive with anything that we've learned about. Come on, let's get my camera working. In terms of genetics. Sorry, getting there. I'll take the batteries, blow out 'em. This isn't that far removed from steam power, to be honest, in terms of current technology. Amazingly, though, that works. So the deal is that these alleles are antigens. And so when you have I'm actually going to do it as a letter. Obviously, they're not letters because that would be kind of silly. If you have the A antigen, right, so these are things that are actually detected by your own immune system. You have the A antigen and no A antibodies. AB stands for antibodies. Probably better write that out in full, actually. Which would be good, because if you had A antibodies, you'd be kind of screwed, right? You'd attack your own blood as being foreign and you would die. So you have the A antigen and no A antibodies. But you will have B antibodies. If you have the B antigen, right, and that would be whether you have 1 or 2 copies of the B allele, but no A allele. You have the B antigen and no B abs and B antibodies. Right, if I just do O down here, O equals no antigens. And A and B antibodies. So if you're an O individual, that's great for everybody else. It really sucks for you though, because it means that you can't accept blood from anybody other than an O type donor. Isn't that crazy? So O is the universal donor because it doesn't have any antigens. So that blood will not react with anybody, right? Because you take only the red blood cells out, you leave all the other stuff behind when you put it into another individual. Right, so if you don't have the antigens, you have the antibodies, which is how my blood cells are getting smaller and smaller. If you have both A and B antigens. So I had to write this in red because, you know, it's blood. That equals no antibodies. So now you have a different phenotype for A dominant and B dominant and AB dominant. So AB individuals are universal receivers. If you have AB blood, you can get blood from anybody. Like some random person in the street, get blood from them, you'll be fine. Obviously, you probably want to, you know, make sure that you don't have any bloodborne diseases and stuff. But if they're all clear, then you can take blood from anybody. If I'm B positive, right? So I have, oops, sorry. That would be potentially confusing. So I have A antibodies in my blood. So I can't get blood from any individual that has an A antigen, right? So I can only receive blood from people that are B or that are O. Otherwise, my immune system will detect those blood cells and kill them. Isn't that cool? So this is how you can have expression of two different traits, is because we're looking now at a molecular level. We're looking actually, and the proteins that are present on the surface of your red blood cells are what determine the phenotype you express in terms of blood type. And that phenotype is really a combination of what antigens you have, and therefore what antibodies you have. So if you have the A antigen, you have the B antibodies, and vice versa. If you have the AB antigens, you have no antibodies. And so you can get blood from anybody. If you're O type, you're kind of, well, I mean, you're not screwed, but you're not in as much of a happy place as AB individuals would be.
So, sir, this
Oh, hang on, Jordan, got a question from Dipper. So in terms of physical differences, yes, there is a physical difference. It's the antigens that are present on the surface of the red blood cells. But to find that out, sorry, dicky knee. To find that out, you'd need to take a sample of blood and separate the red blood cells out and see what antibodies those red blood cells react with. And so if you do an antibody reaction test like an ELISA, pardon me, you would, if you were O type, you would see no reaction to any of the antibodies. So the only other way of figuring that out, if you don't like needles, is again to look at genealogies, right? So if I am B, then I am either heterozygote, B0, or homozygous dominant B. So that would mean my mother and father, because I'm pretty sure this is on the, I would have had to have got a B from one of my parents, potentially another B from someone else. Well, not someone else, not like the milkman or anything, but one of the other parents. So if your mother is A and your father is O, so you will be either O or A. So essentially, this is always fun. I love doing stuff like this. It's very easy to get me distracted on things. So your father will contribute an O allele, right? Because it's homozygous recessive, right? It can only be that. Your mother is A, and so she'll either be homozygous dominant or heterozygous. So, oh yeah, you could just donate blood. That's also another easy way. But this is more fun, although you don't get like a candy or a gift voucher or anything. Yeah, and needles are involved too. You know, I can't give blood because I grew up in the UK during the mad cow disease epidemic, which is very sad. Your mother will contribute either an A or an O if she's AO. Oh my God. Yeah, maybe you're not the ideal blood donor there, Dipper. So yeah, so you're either A because you're heterozygous, or homozygous recessive O, so type O. So you see, it's fun. And then if you have children, or if you will have, or if you have had children, and you find out their blood types, then you could work out which of those you are based also on what your children are. Yeah, so AO is your genotype, right? A is your phenotype, that's your blood type. Yeah, so that's the important distinction. That's why if you're heterozygous AB, you have the AB phenotype. If you're heterozygous AO or BO, you have the A or the B phenotype. It's really weird. There's actually something called compound heterozygosity. So you can have two different disease-causing alleles and have the disease even though you're heterozygote. It's kind of like that, I guess, but not quite. It's a bit different. So yeah, blood type's cool. I love talking about blood type. Jordan, you had a question? I know you're going to ask something before I got distracted with, well, not distracted, went off talking about Dipper's stuff. Speak now, Jordan, or forever hold your peace.
Sorry, I got pulled away real quick. It was
worries.
about the RH factor.
Oh, geez, I don't know
Yeah.
jack about that.
Okay.
In fact, I did actually
enough.
look it up once because another student asked about it, and I got so confused that the next class I was like, yeah, I don't know.
Okay, fair enough. 'Cause the only thing I know
it's-
about it is like it's activated after pregnancy
Right.
in women,
Yeah,
but
yeah,
that's
yeah.
pretty much my limit.
Yeah, same here. I know I'm B positive, but I did look into it, and there's a couple of different ways of describing the trait and the genetics behind it that don't completely agree. And even if you pick one of them, oh, yeah, it hurt my head. There you go. That's probably the easiest way of describing it. When I looked at it on Wikipedia, there's like this huge table of all these different genotypes and combinations which generate either a rhesus positive or rhesus negative. And so I don't actually understand it, is a quick and easy answer. I've tried. Maybe if I sat down in like a dark room for several days, I'd get it. But at first glance, it was way complicated. So yeah, sorry, you're not gonna get not gonna get a good answer from me on that one. So let's do these very quickly down here. So I'm going to jump around. The reason is I want to get these done and then I can have time to talk about epistasis. So one thing that's important to talk about is trying to figure out, this is particularly for polygenic traits, and you have mutations. Is that mutation in the same gene or a different gene in that pathway? Right, and so this is a process called complementation. And so essentially what it means is that if you have two different alleles, right, so you have two different homozygous recessive individuals, right, or two individuals that express the same mutant phenotype, if you cross those individuals together, you should get a heterozygote, right? Now, if both of those alleles are in one gene, you'll get a heterozygote, but you'll still not get function. Does that make sense? Because you will have a mutant allele from both, that compound heterozygosity that I was talking about earlier. And so you will still express, even as a heterozygote, the mutant phenotype, whatever that might be. Even though the parents were homozygous for each different allele. Now, if you do the same thing and you have those mutations are in different genes, right? So here we have homozygous recessive for one mutant allele, homozygous recessive for the other one. Now, when you cross those two individuals together, you will end up with a heterozygote for each gene. So you will have a dominant allele for each gene. Therefore, you will not see the mutant phenotype. You'll see the wild type phenotype. So this is actually a pretty neat trick to show that the mutation that you think is in a particular gene is in that particular gene. You can do this via other ways too. You could add the wild type gene back via like an extra chromosomal array or something, and it should rescue that mutant phenotype. So you have a wild type phenotype again. Jordan.
Sorry, I keep interrupting. That
Questions
goes to
are good.
mostly to the students as well, to everybody else, because I'm dragging this along. So would the O phenotype genotype, would that not just be a lack of the AB antigens, or like I'm assuming they're like proteins or something on the edge of the blood cells?
Yeah, so they're glycoproteins. So they're proteins that have sugars added to them.
So would it just not be the lack of something in that
They just
I don't
don't,
know.
individuals that have the are homozygous recessive for the O allele do not produce those glycoproteins. So they do
Okay.
not, there are no antigens, at least those antigens, present on the surface of the red blood cell.
Okay. Gotcha. Okay, thanks.
And so that's why when you give blood, typically what happens is that they will separate the red blood cells out from the plasma. And if they're not keeping the plasma for anything, they just toss that. And they keep like a nice, like concentrated blood cell mix, right? So they get rid of all of the other stuff, antibodies, whatever, proteins, etc. And they just keep the red blood cells. So those red blood cells are what get put into the recipient. And so what antigens are on those red blood cells is what's important. Because the receiver, the recipient, is going to have those antibodies present. And so if you're an O individual, you will have no antigens on your own blood cells, but you will produce antibodies to A and B antigens. So if you get blood coming in, those red blood cells cannot have any antigens on them whatsoever, either A or B, because if they do, then your antibodies will react with them and bad stuff will happen. So yeah, it's pretty cool. Okay. So a couple of other things, and these are just kind of, these are important terms. Might need to go through epistasis Thursday, because we've got a chunk of time put aside for linkage, and linkage is a real like chunky problem. So we'll be able to use a little bit of that to talk about epistasis, because epistasis is neat. So one thing as well is that even if you have a particular genotype, it doesn't necessarily mean that you'll express the same phenotype, right? So we have kind of two different terms, variable expressivity and variable penetrance or incomplete penetrance. And so if you have, say you're homozygous recessive for, I don't know, a gene in the Krebs cycle or whatever, which would usually be really bad, right? And you do not have any issues whatsoever, even though your genotype is bad, right? You have a homozygous recessive phenotype. If you don't express the phenotype that you would expect from that, then that genotype has less than complete penetrance. Typically, we look at this as a population level too. Right, so some individuals will have the disease, some individuals will not, even if they have the same genotype. I'm just going to write that down. And so that is incomplete penetrance. i.e. the disease phenotype has not completely penetrated through the population so that everybody with that genotype has the disease. Now that's different from variable expressivity. And so that is, and this is often a consequence of other stuff going on, essentially, right? So there are going to be other genes which can modify or ameliorate or make something worse, right? Doesn't necessarily have to be better, it could be also make something worse. And so you can have the same genotype, but different levels of disease. That's called variable expressivity. So that's how strongly you express the disease phenotype. And so that's variable expressivity. And so my favorite example to use for that is actually mitochondrial disease, right? Which is a fairly poorly understood disease, which is basically the result of mutations in your mitochondrial DNA. Hereditary mental illness. Ooh, I mean, that's super complex. That's both a complex polygenic trait, so there are going to be many, many different genes involved in that trait, and it's going to have almost certainly a genotype bioenvironment interaction, where some individuals, depending on their environment, will be more predisposed to that phenotype or disease versus others. Yeah, that would be a very, very complex one to answer. However, also, right, and we'll be talking about this in terms of linkage, sometimes many different genes kind of encoding a trait will be on the same chromosome within linkage distance of each other. And that basically means that if you inherit a allele for one chromosome, one gene, you stand a greater chance of inheriting that, a specific allele for another gene. And so quite often you'll see particular phenotypes, even if they're a result of multiple different genes, passed down kind of as a package through generations. And so there's a lot going on there, Dipper, and I'm really not super well equipped to answer that question because I don't know much about mental disease and the genetics of it. But without a doubt, that's going to be a very complex problem because you're going to have lots and lots of genes involved, without doubt, developmental, maintenance, function, all of that kind of stuff. And also there's the influence of the environment, what you eat, what your mother eats or mother ate while you were pregnant, whether she smoked or drank, whether you smoke or drink, what you eat, what your upbringing was like. Was it stressful? Was it, idyllic? Were you petting baby goats every day or growing up in a war zone? So there's a lot there, and I wouldn't even want to try and tease out what the contributing factors are. Those are very difficult questions in human biology, without a doubt. In fact, most questions are actually really difficult. So yeah, in terms of mitochondrial disease, right, what mitochondrial DNA you inherit from your mother, because you don't get any from your father at all, can have an effect on whether or not you show this disease, right? But also, you have more than one mitochondria in each cell. So if you have a lot of functioning mitochondria and only a few non-functioning mitochondria, you won't express the disease, right? So the penetrance is incomplete, right? Because there's a threshold kind of effect. Also with mitochondria, it depends where those mitochondria are, right? So if you have, you know, duff mitochondria in your hair cells, like not a big deal. If you have a lot of duff mitochondria in your neurons in your brain, that's a really big deal. Or if they're in your muscle, you'll see a much stronger phenotype based on the relative prevalence of functioning versus non-functioning mitochondria. Right, so that's kind of like a cool example to talk about. It's not a very well understood disease, to be honest. They used to call it like the yuppie flu and stuff like that. But there's always it's always the context, right? And that's really like the big takeaway, is that almost all genes, all traits, sorry, are the outcome, what you see in terms of the phenotype is a result of many different genes and their genotypes and their interactions with each other and the context that they're in and the environment as well. And the sum of all of that gives you the phenotype that you can see. Okay, I think that's enough for now. On Thursday, we'll talk a little bit about, yeah, maybe go over gene bioenvironment interaction if that's necessary. Kind of talked a bit about that already, but also talk about epistasis, which is important because it breaks one of Mendel's laws, and we'll be kind of touching on that a little bit later too. All right, I think it's time for lunch. We finished, well, we didn't finish everything, but we finished on time, which is always a plus. And I will see you back here at 2 for the lab, which shouldn't be too heavy. We're just going to introducing the linkage lab. So kind of going over the genetics and the traits that we're going to be working with and some of the assessments that go with that too. So I'll see you in half an hour. See you at 2 o'clock. Thanks for all the awesome questions. It's a lot of fun answering those rather than just me talking at, you know, Robert. Not that's a bad thing, Robert. There's only one of you. So yeah, never be afraid to ask questions. It always makes my life a lot more interesting. Cool beans. I shall see you all in a little bit.